Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. In this lecture, I will be going over DNA replication and DNA repair mechanism. Let's get into the details. Genetic information is transmitted from parent to progeny by replication of a parental DNA, a process in which two daughter DNA molecules are produced that are each identical to the DNA of a parent. During DNA replication, the two complementary strands of parental DNA are pulled apart, and each parental DNA is then used as a template for the synthesis of a new complementary strand. And this type of DNA replication, we refer it as semi-conservative replication. And during cell division, each daughter cell receives one of the two identical DNA molecules. Now let's compare a replication of prokaryotic and eukaryotic chromosomes. Note that the bacterial chromosome is a closed, double-stranded circular DNA having a single origin of replication. A separation of two parental strands of DNA creates two replication forks that move away from each other in opposite directions around the circle. Replication is thus a bidirectional process. The two replication forks eventually meet, resulting in the production of two identical circular molecules of DNA. Whereas in eukaryotic chromosomes, so each eukaryotic chromosome contains one linear molecule of double-stranded DNA having multiple origins of replication. Bidirectional replication occurs by means of a pair of replication forks produced at each origin. Completion of the process results in the production of two identical linear molecules of double-stranded DNA. We refer them as sister chromatids. So the DNA replication occurs in the nucleus during the S phase of the eukaryotic cycle. The two identical sister chromatids are separated from each other when the cells divides during mitosis. So in the figure here, panel A, it shows a eukaryotic chromosome replication during S phase, whereas panel B, it represents different representations of replicated eukaryotic chromosome. Now in this slide, the comparison between DNA and RNA synthesis is shown. So overall, the process of DNA replication requires the synthesis of both DNA and RNA. These two types of nucleic acids are synthesized by DNA polymerase for DNA and RNA polymerase for RNA. Now let's look at the similarities between DNA and RNA synthesis. The newly synthesized strand is made in 5' to 3' direction, both in DNA and RNA, as you can see here in the figure. The template strand is scanned in the 3' to 5' direction, both in DNA and RNA synthesis. The newly synthesized strand is complementary and anti-parallel to the template strand, that is same both in DNA and RNA synthesis. And each nucleotide is added when 3' hydroxyl group of the growing strand reacts with the nucleoside triphosphate, which is base paired with the template strand, and then the pyrophosphate, that is the last two phosphates, are released during this reaction. Now coming to the differences. This includes the substrates for DNA synthesis are the deoxyribonucleotides, whereas the substrates for RNA synthesis are nucleotide triphosphate. So that's DNA synthesis needs deoxynucleotide triphosphates, whereas for RNA it needs just nucleotide triphosphate. So the difference is the sugar, pentose sugar. Now DNA contains thymine, whereas RNA contains uracil. And DNA polymerases require a primer, whereas RNA polymerases, they do not require a primer. That is, DNA polymerases cannot initiate strand synthesis, whereas RNA polymerases can initiate strand synthesis on their own. And the last difference is DNA polymerases can correct mistakes, which is referred as proofreading, whereas RNA polymerases, they cannot correct mistakes. Because DNA polymerases have 3' to 5' activity, that's why they can correct mistakes and do the proofreading. Now here is the table that compares between DNA and RNA polymerases. As you see, a DNA polymerase, the nucleic acid that is synthesized in 5' to 3' direction by DNA polymerase is DNA, and by RNA polymerase it is RNA. And the template requirement for both DNA polymerase and RNA polymerase is DNA there, which is in 3' to 5' direction. And the substrates that is required is deoxynucleotide triphosphates for DNA and nucleotide triphosphates for RNA. The primer requirement here for DNA polymerase and RNA polymerase. So here for DNA polymerase, primer required is DNA or RNA, whereas for RNA polymerase, there is no primer required. And also the required template that has got 3' to 5' exonuclease activity, DNA polymerase has that, whereas RNA polymerase do not have it. Now let's move on to see steps of DNA replication. So at the origin of replication, helicases, they will bind to a DNA strand and unwind the double-stranded DNA and allow the free single-stranded DNA to be available for replication. So a single-stranded binding proteins will prevent a reannealing process. Now DNA replication we can study in leading strand synthesis and lagging strand synthesis. Now the leading strand synthesis, it's a continuous process, whereas lagging strand synthesis is a discontinuous process. So the leading strand synthesis, here a DNA primase synthesizes the primer in 5' to 3' direction, and DNA polymerase extend the primer moving into the replication fork leading strand while leading strand is synthesized. And while helicase, it continues to unwind the DNA as it is shown in the figure. Coming to the lagging strand synthesis, so the primase synthesizes the primer which is in 5' to 3' direction, and DNA polymerase extend the primer moving away from the replication fork. This is what happens in the lagging strand synthesis. And the synthesis stops when DNA polymerase encounters the primer of the leading strand on the other side, or the primer of the previous 𝗒𝐫𝐫𝐞 fragment. A helicase opens more of the replication fork, a third 𝗒𝐫𝐫𝐞 fragment will be added, and then RNase-H, that is a 5' exonuclease activity containing molecule, to digest the RNA primer from the fragment. So in the eukaryotic cell, DNA polymerase, it extends the next fragment, that's fragment number 2, to fill in the gap. Whereas in the prokaryotic cell, DNA polymerase 1 has both 5' exonuclease activity to remove the primers and the DNA polymerase activity to extend the next fragment, that is fragment number 2, to fill in the gap. So in both types of cells, DNA ligases connects fragment number 1 and 2 by making a phosphodiester bond. This whole process repeats to remove all RNA primers from both leading and lagging strands. So this is what happens during DNA replication. So all those points that I have written here in brief. So origin of replication, and then helicases separates DNA strands, and then single strand DNA binding proteins will prevent reannealing process, and primerases, that RNA primers are synthesized in 5' to 3' direction, and then the DNA polymerase 3, which basically it extends that in 5' to 3' direction. So making a leading strand which is a continuous process, whereas lagging strand is a discontinuous process using 𝗒𝐫𝐫𝐞 fragments. And then the primer removal and gap filling happens, and in eukaryotes, RNase-H, which are uncharacterized DNA polymerases, will do that, whereas in prokaryotes, DNA polymerase 1 does the both. And then proofreading, it is present in DNA polymerase, like DNA polymerase via its 5' to 3' direction, nuclease activity will do the proofreading process, and then DNA ligase seals the nicks between the 𝐒𝐫𝐤𝐾 fragments in the lagging strand, and DNA 𝐜𝐫𝐼𝐬𝐺s, that is DNA topoisomerases, provides a swivel in front of each of the replication fork as the replication continues, and it opens up the double-stranded DNA, loosen the heterochromatin to make it as a euchromatin and basically allow the replication to move on. Now, eukaryotic DNA polymerases. So, DNA polymerases alpha and delta, they work together to synthesize both the leading and lagging strand, whereas DNA polymerase gamma, it replicates mitochondrial DNA. And DNA polymerase beta and epsilon are thought to participate primarily in DNA repair mechanism, and DNA polymerase epsilon may substitute for DNA polymerase delta in certain cases. So, in this table, a DNA replication between prokaryotic cell and eukaryotic cell has been compared, as you see here. So, all the details are written here, including the steps in replication and what happens in the prokaryotic cell and what happens in the eukaryotic cell. Now, the site of replication, that is ovary. So, a origin of replication, that is referred as ovary. So, one ovary site per chromosome is seen in prokaryotes, whereas in eukaryotic cell, multiple ovary sites for chromosome is seen. Unwinding of DNA double helix is done by helicases in both prokaryotic and eukaryotic cell, and then stabilization of unwounded template strands done by single-stranded DNA binding proteins in both the type of cells. Synthesis of RNA primers are done by primerases in both the cells. Synthesis of DNA leading strand and the lagging strand, like it is done by DNA polymerase 3 in prokaryotic cell, whereas DNA polymerase alpha and delta eukaryotic cell. Then the removal of RNA primers done by DNA polymerase 1 having 5' to 3' exonuclease activity in prokaryotic cells, whereas reannease H is the one that will remove RNA primers in eukaryotic cell having 5' to 3' exonuclease activity. And then the replacement of RNA with DNA in prokaryotic cell done by DNA polymerase 1, whereas in eukaryotic cell it is done by DNA polymerase delta. And then joining of 𝗒𝐫𝐤𝐾 fragments is done by DNA ligases in both. And removal of positive supercoiled side of advancing replication fork, it is done by topoisomerase 2, referred as DNA gyrase in prokaryotic cell, whereas in eukaryotic cell it is also done by DNA polymerase 2. Synthesis of telomeres not required in prokaryotic cell, whereas it is done by telomerase in the eukaryotic cell. Now, certain drugs will target topoisomerase enzymes. So, those are quinolones, which are levofloxacin, ciprofloxacin, moxifloxacin and ofloxacin. So, they block action of prokaryotic topoisomerase 2, that is DNA gyrase. Nalidixic acid kills bacteria by inhibiting DNA gyrase. Etoposide, tenoposide inhibit eukaryotic topoisomerase 2 enzyme and they act as anti-cancer cell. These are some of the drugs that target topoisomerases during DNA replication process. Coming to telomeres, role of telomerases. Telomers are repetitive sequences at the end of linear DNA molecules in eukaryotic chromosomes. With each round of replication in most normal cells, the telomeres are shortened because DNA polymerase cannot complete synthesis of the 5' end of each strand. This contributes to the aging of cells because eventually the telomeres become so short that the chromosomes cannot function properly and the cells die. So, the answer to this shortening of the telomere is telomerase enzyme. Telomerase is an enzyme in eukaryotes used to maintain the telomeres. It contains a short RNA template complementary to DNA telomere sequence, as well as the telomerase reverse transcriptase activity. So here in the figure, as you see, the telomerase enzyme is shown. So it is going to elongate the telomere at the ends of the chromosome. So the telomerase is thus able to replace telomere sequence that would otherwise be lost during replication. And normal telomerase activity is present only in embryonic cells, germ cells, which are like reproductive cells, stem cells, but somatic cells do not have a telomerase enzyme activity. However, cancer cells often have relatively high levels of telomerase, preventing the telomeres from becoming shortened and contributing to immortality of malignant cells. This is the information that you must remember related to telomeres and telomerase enzymes. Let's look at reverse transcriptase enzyme activity. Now, the reverse transcriptase is an RNA-dependent DNA polymerase enzyme that requires an RNA template to direct the synthesis of new DNA. Most notably, human immunodeficiency virus uses this reverse transcriptase enzyme to replicate their RNA genomes. Now, the DNA synthesis by reverse transcriptase in retroviruses can be inhibited by AZT, DDC and DDI. These are the drugs. Now, eukaryotic cells also contain a reverse transcriptase activity, like it is associated with telomerase, like human telomerase activity, encoded by retrotransposons, which are residual viral genomes permanently maintained in the human DNA, and these they will play in amplifying certain repetitive sequences in DNA molecule. Now let's look at DNA repair mechanism. The structure of DNA can be damaged in number of ways through exposure to chemicals or radiation. Incorrect base can also be incorporated during replication. Multiple repair systems have evolved, allowing cells to maintain the sequence stability of their genomes. If cells are allowed to replicate their DNA using a damaged template, there is a high risk of introducing stable mutations into the new DNA. Thus, any defect in DNA repairs carries an increased risk of cancer. Most of the DNA repair occurs in the gap1 phase or the G1 phase of eukaryotic cells, and the mismatch repair mechanism occurs in the G2 phase to correct replication errors. So here is the table that gives or that summarizes different types of DNA repair mechanisms. That thymine dimer repair mechanism, mismatched base pair repair mechanism, and then we have cytosine deamination repair mechanism going on. So cause given for each one of them. Thymine dimers are because of UV radiation, mismatch base is because of DNA replication errors, or cytosine deamination may be spontaneous or exposure to heat. And the excision or replication enzymes here, like excision endonuclease, where thymine dimers is the one that is taken care and which is deficient in xeroderma pigmentosa case. And repair enzyme there is RNA polymerase and DNA ligase. So like this for each mismatch repair mechanism, so details are given here. Now let's look at repair of thymine dimers. Ultraviolet light induces formation of dimers between adjacent thymines in a DNA. So thymine dimers interferes with the DNA replication and normal gene expression. So a nucleotide excision repair mechanism eliminates these thymine dimers and allow DNA replication to go on. Now how exactly a nucleotide excision repair mechanism will go on? So it is shown in the figure below here. An excision endonuclease, that is sometimes called as exonuclease, it makes a nick in the phosphodiester backbone of the damaged strand on both sides of the thymine dimer and removes the defective oligonucleotide. And then the DNA polymerase fills in the gap by synthesizing DNA in the 5' to 3' direction using the undamaged strand as the template. and then DNA ligase seals the nick in the repaired strand, and that's how a nucleotide excision repair is done. Now coming to base excision repair mechanism, or which is done during cytosine deamination process. Now the cytosine deaminase is a loss of an amino group from cytosine that converts cytosine into uracil by a uracil glycosylase enzyme. Subsequently, this area is recognized by an AP endonuclease that removes the damaged sequence from the DNA. And then DNA polymerase fills in the gap, DNA ligase seals the nick in the repaired strand. And that's how the base excision repair mechanism is done. This figure, it gives a summary of important genes involved in maintaining DNA fidelity and where they function in cell cycle. As you see here, G0 phase where thymine dimers, a bulky lesion repair going on, and it can be defective in xeroderma pigmentosa, and then the nucleotide excision repair mechanism is going on there. And then we have a DNA polymerase proofreading during DNA synthesis going on with the S phase, and then mismatch repair mechanism in G2 phase. So these are some of the things that will summarize the cell cycle and the repair mechanism going on in each phase of the cell cycle. Now let's move on to see diseases associated with DNA repair. Inherited mutations that result in defective DNA repair mechanisms are associated with a predisposition to the development of cancer. So one of the disorder in that respect is xeroderma pigmentosa. This xeroderma pigmentosa, it is an autosomal recessive disorder characterized by extreme sensitivity to sunlight, skin freckling and ulcerations is seen, and also high risk for skin cancer is seen in this particular disorder. The most common deficiency that is seen in xeroderma pigmentosa is the 𝐒𝐫𝐞 enzyme deficiency. So patients should avoid exposure to any source of ultraviolet light to prevent themselves from skin freckling, ulcerations, and eventually getting into development of skin cancer later on. The other disease that I would like to go over here is hereditary non-polyposis colorectal cancer, which is referred as Lynch syndrome. So here mutation in the genes, that is MLH1 and MSH2 is seen. So the defect in DNA mismatch repair mechanism is the cause for this particular disorder. Microsatellite instability is prominent type of error that is seen here, and loss of mismatch repair function is seen, and retention of errors may lead to cancer progression in hereditary non-polyposis colorectal cancer. Now the mutation or deletion of tumor suppressor genes can also be seen, and here one of the such disorder is Leigh-Fraumeni syndrome and many solid tumors where there will be inactivation of a tumor suppressor gene, that is P53 gene. Coming to ataxia telangiectasia. So here a mutation in ATM gene leads to inactivation of ATM, and that leads to hypersensitivity to X-rays and predisposition to lymphomas. And also mutation in BRCA gene in the breast cancer, prostate and ovarian cancer, and mutation in BRCA2 is noted in breast cancer. Retinoblastoma gene, that is RB gene, it's a tumor suppressor gene, and it's a negative regulator of cell cycle. Its ability to bind the transcription factor E2F and repress transcription of genes is required for the S phase, and the mutation of this gene will lead to retinoblastoma. So this is all about DNA replication and repair mechanism. Thank you.